What you're going to hear today is a result of a collaboration between myself and Bruce Deamer, who's here in the audience. Bruce and I have come up with a new idea about how life began on this Earth and perhaps on Mars. And that's what you're going to hear about from me. And on the way, we're going to talk about a new technique for analyzing DNA that was developed here at UC Santa Cruz. Can I have the first slide? And let's push this. And there it is. I had a new idea. Hello? There it is. I had a new idea about 30 years ago now that has developed into a way to analyze DNA right in terms of single molecules passing through a pore. And I want to start out with that so that you can understand the rest of what I'll be talking about. Here is a notebook dated June 25th, 1989. And here you can see a pore, and that little thing going from minus to plus is a strand of DNA. Under the pore, you can see four bases of DNA: cytosine, guanine, thymine, and adenine, CGTA, and each base gives a different electrical signal. Well, that was just an idea back then. 30 years later, you're going to hear about how that idea has developed. I was a freshman in high school in 1953 when Watson and Crick came out with a structure of DNA. It was a beautiful double helix, and I was fascinated by it. I even memorized the actual name of deoxyribonucleic acid, DNA. And that has stuck with me all my life, that moment of seeing that structure. It's also a part of your lives. DNA guided the process by which everyone in this room grew from a tiny fertilized egg to 100 trillion cells of the human body. So keep that in mind. The DNA in every living cell in your body is about this long if we line it up end to end. And written into that DNA are the 20 to 30,000 genes that guide us. Even today, we're being guided by those genes, and certainly they guided us as we grew to maturity. So what is DNA, and where did it come from? Well, I need to pass along a couple of facts, because if I don't tell you about these facts, you won't understand the rest of this. Life requires water. That's easy. Everybody knows that. Everybody knows that there's two kinds of water in our world. There's a salty water of the ocean that just laps up to our beaches, and there is the fresh water that falls as rain. And that water evaporates from the ocean and falls as basically distilled water, falls on land masses all over the world. So if life requires water, where did it begin? Was it begin in salt water or in fresh water? Well, everybody says salt water is 99% of the water on Earth, so it's obvious that life must have begun down in the ocean somehow. Nobody knows how. So that's been the paradigm for the last probably 50 years, I would say. Everyone except Charles Darwin thought that. Charles had a different idea back in 1871 that he wrote about to his friend Joseph Hooker. I'll just read this to you. But if, and what a big if, we could conceive in some warm little pond with all sorts of ammonia and phosphoric salts, heat, light, and electricity present, that a protein compound was chemically formed, ready to undergo still more complex changes. That little idea of a warm little pond has been in the back of everybody's mind, but he was thinking of a pond of fresh water. Life began 4 billion years ago, we think. Were there warm little ponds back then? Bruce and I put together a video showing what we think the world was like 4 billion years ago. What you see is that it's lifeless. There's nothing green there because plants had not yet started up. There were geysers, just like Yellowstone, if you visited there. The water from the geyser runs downhill, and the brown stuff on top is organic compounds that are floating in the surface of the water. As the water flows downhill, it makes little puddles, and the most important thing about those puddles is that they're not just stationary. They dry out, they get wet again, they dry out, they get wet again, and that wet-dry cycle, I think, is very important to the story that we're going to be telling you. So I wanted to see this for myself, and for the last 20 years or so, I've been wandering around to various volcanoes on Earth. Iceland, for instance, is basically a big volcanic island. Hawaii, of course. Even here in California, we have an active volcano called Mount Lassen. And some of you may have visited there and perhaps even visited Bumpus Hill, where Bruce and I have been doing some of our work. So keep that in mind, because I went to Kamchatka in 2004 on a field trip. And here I am on Mount Matnovsky in southern Kamchatka. Kamchatka is basically in Russia. It's far eastern Russia. And this is about a day's drive from a little city called Petropavlovsk, that is the main city in Kamchatka. And I did kind of a silly experiment, but it taught me a lesson. The silly experiment was to find not a warm little pond, but what we call Darwin's hot little puddle. And into that puddle, I poured a prebiotic soup, containing 4 amino acids, 4 nucleobases of DNA and RNA, a carbon compound called a fatty acid, which is basically a soap, and that's going to become important, so remember that. Also some glycerol, phosphate, and the pool was a pH 3, which is it's mildly acidic. It's about like lemon juice, okay? Then, over a period of minutes and hours and days, we took samples of that pool and brought it back home to analyze in the lab. But one thing that I noticed immediately is that around the edge of the pool, a white froth had appeared. That shouldn't be too surprising. I put a soap in. Of course, that soap is going to form that froth. But I know for a fact that that froth is made up of membranes, self-assembled membranes. And everyone sitting here, once in your life or more, has in fact made a self-assembled membrane because we have all had fun blowing soap bubbles. Soap bubbles are an example that everybody's experienced of a self-assembly process. There is no physical laws that really tell a soap bubble how to appear. It just happens. We call that an emergent phenomenon. And so I had the idea that maybe life emerged here, okay? Because those little membranes contained the other stuff that I put into the pool. Now then, there's something else that's quite important for you to understand, and that is the process that we call self-assembly into polymerization. Now, polymer it's a chemical term, but you all know polymers because polyethylene, polystyrene, polyesters, you're probably wearing some polyesters in the clothing that we have on. Those are all polymers. Poly means many, and they're composed of monomers. Mono means one. So the units of a polymer are called monomers. The monomers of nucleic acids are called nucleotides. There's 4 of them in DNA and a slightly different 4 in RNA. So those 4 are A, C, T, and G that I showed you in that initial little sketch. Okay, let's now take a look at what happens when lipid self-assembles into membranes under the microscope. This is what I see. Over here to the left is dry lipid. Over here is water. And as the water penetrates into the dry lipid, this wonderful set of tubular structures and membranous vesicles just appears like magic. And they actually move like that. That's about the motion that we can see under the microscope. It looks alive, but it's only one little part of life. It's a self-assembled part of life. Okay, now you know about polymers and you know about self-assembly, so I'm ready to go to the next step. And that is, what is DNA really? This is the double helix of DNA that Watson and Crick worked out so many years ago. And I told you it is a polyester. And sure enough, every red molecule that the arrows point to is part of an ester bond. So if I want to make DNA, all I have to do is to link together the phosphate, that's the yellow one there, the sort of orange color, the phosphate and what is called a hydroxyl group on ribose or deoxyribose. And that's an ester bond, and that's what holds DNA together in our bodies. Okay, how to do that? I came back to Santa Cruz and built a simulation chamber. I'm not going to show it to you. It's about so by so. It has carbon dioxide atmosphere, and I could put little samples of the mononucleotides and lipids of DNA through wet-dry cycles, just as we saw in the volcanoes. And look what came out the other end. We see cellular structures. That's the lipid that you see here. But look inside. That is the DNA that we have synthesized by nothing more than wet-dry cycles, simulating what we see on volcanic puddles, those hot little puddles I showed you. So this now has turned into a new idea. I'm going to show you what that looks like. So Bruce and I have traveled to places like this. We've been up to Bumpus Hill. We've been to Australia. Elsewhere, we're going to go down to New Zealand in February of next year, do some field research on this. So we really have gotten a sense of what volcanoes are like. And what we think happened in the origin of life is that there was a synthesis of organic compounds that were delivered to volcanic landmasses where they accumulated. They were flushed downhill into hot springs where they accumulated. And here is one of those hot springs going through a wet-dry cycle. Wet, dry, wet, dry. We have protocells in the wet. We have the synthesis of polymers in the dry phase. Those then drift downhill into other pools. They're now evolving, and they're adapting to increasingly salty water, and the salt water finally turns into a niche that they can occupy. These little guys here are called stromatolites, and we've seen those, Bruce and I, in Australia, 3.5 billion years old. They're the oldest evidence of life on Earth. Okay, so that's the story. I want to tell you one last thing, and that is that Mars, 4 billion years ago, also had shallow seas, and it has volcanoes. We still see the remnants of volcanoes on Mars. So Mars had just the properties that I've told you about that we think were on early Earth. Because Mars cooled off first, we think that maybe, just maybe, these processes that I've showed you started there before they began on the Earth, which was still too hot and did not have an ocean. So keep that in mind, because I'm going to change the subject slightly and get back to that first sketch I showed. Here is a nanopore sequencer. It's called a MinION. It's done by a British company, Oxford Nanopore Technology. They licensed the patents that we developed here at UC Santa Cruz. This little guy, you plug it into your laptop, put your DNA in the little port there, and you begin to see the DNA-based sequences appearing on the screen. This is so small. You know, I just carried it here. This is about $1,000 if you want to buy one. They're for sale. This traveled all the way over Africa and followed the Ebola epidemic. And every 90 minutes, one flies overhead in the International Space Station. Let me show you that. Here's Kate Rubin. It's about 2 years ago. She was floating around in the International Space Station, and she used a MinION, there it is, to sequence DNA on the space station in microgravity. The reason to do that, from NASA's perspective, is the space station is not sterile. It's got a film of bacteria and fungal spores all over the inside. If any of those became pathogenic, the MinION would immediately tell the people on the ground what the astronaut was suffering from. So this TEDx is hope. It's about hope. And you know what I hope? I hope that sometime in the near future, in the next 10 to 20 years, we're going to have people on Mars, going to have humans on Mars. And I hope that an astronaut like Kate Rubin might bring along something like that Minion, melt a little ice, and see if there's any DNA left over from the organisms that might still either be alive or extinct and left their DNA behind. If she can do that, and if the sequences in the DNA match the sequences that are known to be part of biological organisms on the Earth, we might have fun thinking that maybe, just maybe, life began on Mars and that was delivered to Earth so that we can trace our ancestry of all life on Earth back to Mars, including you and me. Thank you.